Okay, so welcome back to this next video in which we are discussing cystic fibrosis and cystic fibrosis medications. Okay, so we're in the process now of discussing the origin of the symptoms of cystic fibrosis. So we want to understand, why does having a double knockout of the cystic fibrosis transmembrane conductance regulator, okay, and we're imagining that it's the delta F508 mutation that has happened to both of our CFTR genes. Why does that result in the symptoms of cystic fibrosis? Now, we've discussed now why it results in your sweat being too salty. Okay, what we want to move on to now is discussing the lung disease that it causes, okay, and this is generally what kills people with cystic fibrosis nowadays. Okay, right. So, the lung then. We're going to start off with some background physiology and anatomy and that sort of stuff, so that we can then understand what's going to happen in the case of cystic fibrosis. So just like we did with the sweat gland. Okay, so what I'm going to start off with then is the bronchial tree. Okay, so just some basic anatomy then. Okay, so let me firstly draw then the trachea there, and is this at the right height? There we go. So this is representing the trachea here, okay, also known as the windpipe. Now what happens then is, as the trachea comes down, it then splits into two separate branches, okay. Here is the left bronchus, sorry, not the left bronchus, the right bronchus, okay, patient's right, and we're looking from the front here, okay, and then here is the right, sorry, no, this one's the left bronchus, made a complete pig's ear of that. Okay, so this is the left bronchus here, okay, and then the other one will be the right bronchus. Okay, and really what I want to get across in this picture is just the concept that they split and split and split and split. So if we do it for the right bronchus here, what will happen is this will give off loads of different branches. Okay, here's another branch, here's another branch. Okay, I'm drawing these kind of the way it actually anatomically appears, not just making it up. Okay, and then these branches will branch again. Okay, you'll have one like that, and then another one going off behind. Okay, this one will branch off many different times. Okay, so what my point here is that the bronchi split and split and split and split. You start off with one trachea here, and it then bifurcates into the right and the left bronchus. Then each of those split down, and they will continue splitting again and again and again, making smaller and smaller bronchi. Okay, then as the bronchi get smaller and smaller, they become known as bronchioles. Okay, and then finally you'll get down to bronchioles, which are so small that they actually have the alveoli coming off them. Okay, so let me show this now. So here is a bronchiole that is so small that it's actually got alveoli coming off it. So here is an alveolus coming off this bronchiole. Here is another alveolus. Here is another one. Okay, and these will be obviously cells lining these, but they'll be very, very thin cells. Okay. Then we might have another one here, another one here, okay, another one here, like so. So these are alveoli. Okay, and the alveoli will be surrounded by a very rich capillary network, which will be taking oxygen from these air spaces into the blood, okay, and then delivering it back to the left ventricle. So this is an alveolus, that's the singular, alveoli is the plural. Okay, right. Now, what I want to do is now take a cross-section down some big bronchus, okay? So let's say I'm going to take a cross-section down this one. So what I'm going to do is I'm going to cut through it, okay? And then I'm going to look at the exposed cross-section and have a look at its structure, okay? Because what we need to understand is what the submucosal glands are, because these are the things which are going to be affected by cystic fibrosis. And I should probably say at this point that the origin of the lung disease in cystic fibrosis is still controversial. everyone agrees on what happens in the sweat glands, everyone agrees on what happens in the pancreas. What happens in the lung is still very controversial, and there are loads of different theories of what happens, and some of them are compatible and some of them are actually incompatible with each other. Okay, I am going to present you the leading theory, okay? The one which you'll find in the textbooks is the one that I'm going to present to you. But I should say that this is still controversial, okay? So it may well be wrong, but it's probably not wrong. Or at least it might be incomplete, is probably a good way to say it. There might be other little parts of the story that are missing from the model that I'm going to show you, okay, which can be added on to it, okay? So what I'm going to tell you about is how cystic fibrosis, the loss of function of the two CFTR genes, is going to result in dysfunction of the submucosal glands in the lung, and this is going to produce very thick, sticky mucus, which is then going to break the mucus escalator, okay, and that's then going to lead to the recurrent pulmonary infections. But despite the fact that that is the leading model and that is the one that's put in textbooks, it is not undisputed, okay? So I just want to mention that, put that out there, that it isn't set in stone, whereas some of the other causes of the symptoms of cystic fibrosis, such as what happens in the sweat gland, is much more set in stone. You won't find people disputing it. Okay, right. So let's have a look then at the structure of the wall of the bronchus so that we can see where these submucosal glands are. Okay, right. So we're now taking a cross-section then through a bronchus, and we want to see what we're going to see. Okay, so the first thing I'm going to draw then is the epithelial cells that line the bronchus. Okay, so of course it's sensible to start with a circle. Okay, and then we're going to have the epithelial cells lining the bronchus here. Okay, now they are columnar epithelial cells, so they're very tall, and they will have cilia. Okay, little projections from their apical membranes that project into the lumen of the bronchus. So these are the cilia here. Okay, and I won't draw them on every single one, but all the epithelial cells will have cilia on them. Okay, so here are all of our columnar epithelial cells here. Okay, right. Now, interdispersed amongst all of these columnar epithelial cells that I'm currently in the process of drawing, you're also going to have some other cells that are not columnar epithelial cells. Okay, now I'll colour these ones in green. Okay, so here in green, these are not going to be columnar epithelial cells. These are going to be what are known as goblet cells. Now, goblet cells are going to secrete something known as mucin. Okay, so I'll firstly just label them up and then I'll tell you what mucin is. Okay, so goblet cells are going to be secreting something called mucin. Now, mucin is what's known as a glycoprotein. Okay, now what that means is it's a protein, a polypeptide, that has had large sugar portions added on, so carbohydrate molecules stuck onto the side of the protein by covalent links, basically. Okay, so that's what's meant by a glycoprotein. Now, mucin is an important glycoprotein. It's the main component of mucus. Okay, mucus is a fancy word for snot and phlegm and sticky goo, basically. The goo that lines the surface of the lungs. Okay? Now, what makes mucus sticky and gooey? It's the presence of the mucin within the solution. So mucus basically starts off as just salty water. Okay, it starts off as water with salt in, and then you stick mucin into it, and that turns it into mucus. Okay, so mucin is the active ingredient, basically, in mucus. Okay, so we will come back to this in a bit when we want to discuss the mucus escalator. Okay, so what's still lacking from my picture? I haven't yet labelled up the fact that these are columnar epithelial cells, which have cilia. Okay, so this is a columnar epithelial cell, and I'll just add some cilia onto this one specifically, so these little projections into the lumen are cilia. Okay, right. Now actually I think we'll discuss at the moment. What's going to happen is you are going to have mucus lining the surface of the lung, okay? So it will be on top of the cilia, it will also be in the spaces in between the cilia, so on the surface of the lungs you'll have this mucus, okay? And remember, mucus consists of the mucin, the glycoprotein secreted by the goblet cells, also in a salty water. And this is important. This is really important. We have not seen yet where this salty water comes from. The other key component of mucus, the fluid, where does that come from? That's going to come from structures called the submucosal glands, okay? And it's the production of the water that is going to go wrong. The fluid of the mucus is going to go wrong in cystic fibrosis, and you're going to end up with a far too viscous mucus because you still secrete the mucin, but you don't secrete enough fluid, okay? So you end up with far too concentrated mucin, and that produces a really thick, really sticky mucus that can't be cleared. Okay, right. So on the surface of the lungs you have the mucus, basically. So this green stuff, this is representing mucus. Okay, right. And I might as well talk about the mucus escalator now, okay? So how do you keep the linings of the airways clear? How do you, you're breathing in horrible stuff all the time. If you go into a field full of cows, you're breathing in all sorts of horrible bacteria and potentially parasites. If you go into the city and you ride your bicycle behind a bus, you're breathing in all sorts of horrible pollution. What keeps the lungs clean of all of this dirt? Okay, well, it gets trapped in the mucus, and then the cilia, these structures here, are going to waft, okay? They're going to move. They're going to gradually move the mucus up and out. So going back to this picture, the mucus will follow a pathway to get it out. The cilia beat so that the mucus moves up the airways and out, okay? So things that get caught in the mucus are going to be moved with this mucus escalator, as it's called, because we're moving the mucus up. That's why it's called an escalator. And then eventually it will then be swallowed, and it will go down the other pipe, the esophagus, okay, and into the stomach, and it will be destroyed in the stomach. Okay, so that's how you keep the airways free of horrible dirt, particles, and bacteria. Okay, now, we're starting to then get a picture here. If this mucus becomes too thick and too sticky that it can't be moved by the cilia, they don't have the power anymore to move it, then it remains stuck there, and you can't clean your lungs. You can't keep your lungs clean. Okay, and I want to stress how important this system is. This is a fantastically important part of the innate immune system, keeping your airways free of bacteria, free of horrible dirt particles and pathogens. This is fantastic at stopping infections in the lungs, okay? It's not your T cells and B cells that are doing this. This is the mucus escalator that is keeping your lungs free of infection, okay? And when this breaks, it's going to lead to real problems, as we'll see later on. Okay, right. So, going back to the structure of the bronchus then, we want to see these submucosal glands. Okay, so let's look beneath these columnar epithelial cells, and I did write cilia at some point. So, the columnar epithelial cells are going to be sitting on a basement membrane, and I can never resist the temptation to put in the basement membrane as a layer. So, in turquoise there, this is the basement membrane, so this is what the epithelial cells are attached to and what they're sitting on. Okay, so this is the basement membrane, and it's a meshwork of proteins, mainly collagen, other proteins like laminins, a bit of fibrin in there as well. Okay, right. Underneath then the basement membrane, what you have is an area of connective tissue that is known as the lamina propria. Okay, so I firstly think I'll show the boundary of this before I then colour it in. Okay, so underneath the basement membrane you have more connective tissue, more collagen and other connective tissue filaments, okay, here in orange. Okay, so this is a layer known as the lamina propria. We're not really going to talk much about this layer. Okay, so that's the lamina propria. I'll label this up. Then surrounding the lamina propria, what you then have is a layer of smooth muscle cells. Okay, so how am I going to draw this? So I'll only make this thin because I'm running out of space. Okay, like so. So you've got smooth muscle cells in this layer, and I'll draw a smooth muscle cell in a moment. Oops. Okay, right, so here I'll put a smooth muscle cell. So remember, smooth muscle cells have this spindle shape where they have a thick belly in the middle here, okay, and then thin edges. Okay, so this is a smooth muscle cell, which for short are abbreviated as SMC. So this stands for smooth, that's the S, then M, that's muscle, and then C is a cell. Okay, and these smooth muscle cells are going to form rings around the bronchus. Okay, so what will happen is this one will be connected to another smooth muscle cell like so. Okay, and then you'll continue round. This one will then be connected to another one, and they'll form huge rings of smooth muscle cells. And when these smooth muscle cells all contract, they all decrease in length. And that means that the entire ring is going to decrease in circumference. And when a circle decreases in circumference, of course, what happens is the diameter decreases in circumference. So, of course, that will constrict the bronchus when these smooth muscle cells contract. Okay, right. So that's the smooth muscle cell there. Okay. Underneath the smooth muscle cell there, you then have the submucosal gland there. Okay, and I've put that label exactly where I want to now put more, so I'm going to have to make the submucosal gland there very small as well. Okay, so around the smooth muscle cells you then have another layer which contains a lot of connective tissue. Okay, so I'll colour this in blue. Okay, and amongst all of the connective tissue you're going to have glands, so I'll leave a space where I can show one of the glands. Okay, so what do these glands look like? Well, they're going to look like a tube, basically. Okay, and that's because they are a tube. Okay, so we're seeing one in cross-section at the moment. We're seeing one of these tubes in cross-section. They'll be lined by, again, epithelial cells, which will be tall epithelial cells, cuboidal epithelial cells, verging on columnar epithelial cells, but they will not, repeat, they will not be ciliated. That's the important difference between these cells here, which are very tall but ciliated, and these ones. These ones are also tall, not quite as tall as these ones, okay, but they're not ciliated. That's the important difference. Okay, so what shall I colour these in? I'll colour these in orange. So here we have loads of epithelial cells lining a tube. Okay, and this entire thing then is called a submucosal gland that we are seeing here. Okay, and this whole layer here, which is known as the submucosa, okay, this is full of submucosal glands. Okay, and these submucosal glands are going to be important in secreting the fluid that is going to make the mucus. Okay, so remember there are two important components of the mucus. The mucin, which is secreted by the goblet cells, and actually it's also secreted by certain cells of the submucosal glands, as we'll see in a moment. Okay, and the fluid, you need the fluid, okay, the water with the salt in as well is important, okay, and it's the fluid that these submucosal glands are going to be responsible for producing. Okay, right, so we now have seen the submucosal glands. Just for a complete picture, I will put the final layer of the bronchi on. We're not going to need this, okay, but just to complete the picture. You'll also have a layer which will consist of sort of fragmented portions of hyaline cartilage. Okay, so I'll put this here. Okay, so these are tough portions of connective tissue. Hyaline cartilage. Hyaline, remember, means that it has a sort of glassy appearance. Okay, so I have the hyaline cartilage here in purple. It's not a full layer of hyaline cartilage. Okay, it doesn't completely surround the bronchus. Okay, you're just going to have fragments of it here and there, which toughen up the surface of the bronchus. Okay, right. So now what we want to do is have a look at the structure of these submucosal glands in more detail. Okay, the first thing I need to tell you is that they do connect up to the surface of the bronchus. At the moment these look like fantastic little tubes that are in the deep layers of the bronchial wall, but what bearing do they have for the bronchial surface? What bearing do they have for the mucus on the bronchial surface? Okay, that's what we now need to see. So these things do actually empty out onto the bronchial surface. Now let me show you one of these in full. Okay, so let me draw the epithelium of the bronchus here. Okay, so I'm showing the bronchial epithelium here, and it's curving in to form a submucosal gland here. Okay, so these are all these columnar epithelial cells that line the bronchus here. Okay, and they'll all be ciliated. So this is just the lining of the bronchus here. Okay, and then for a while, when you go into the submucosal gland, you'll also have ciliated epithelial cells here. But then very quickly, you'll lose the cilia. Okay, like so. So these are the cilia that I'm showing here. Okay, then as you go down, it will gradually split apart, like so, into multiple different tubes that are now in this deeper layer. Okay, so this tube here, okay, which is known as the collecting duct. Okay, so this tube is known as the collecting duct for the submucosal gland secretion. Oh dear, and this hasn't been visible. I do apologise. Right, let me bring this up. Okay, so this is the collecting duct here. Okay, and this is where we're going to be collecting the secretion of the submucosal gland. Okay, then you get up to the top here. This portion is known as the ciliated duct, where you have epithelial cells which actually have cilia on them. Okay, so I'll underline ciliated duct in turquoise. That's this portion here. Okay, then we have the collecting duct here. Okay, which is just lined with columnar epithelial cells, but not ciliated. Okay, so they're tall epithelial cells like so, but they're not ciliated. Okay, and then it's going to split apart into smaller tubes here, which I'll show like so. So these are both tubes here. Okay, one tube here and one tube here. Okay, and these are the tubes that are actually deep in the wall of the bronchus now. So here is the epithelium here. Okay, this ciliated duct and collecting duct, this was the portion that was penetrating down through the lamina propria here, okay, through the smooth muscle cell there, and then we got into the submucosa here. And now these tubes are in the submucosa here. Okay, right. Then what's going to happen is these tubes are going to have other little tubes coming off them, of course. Okay, and then they will end with little structures like so. Okay, and I'm going to have to draw this lining a little bit thinner now. Okay, those cells, of course, should be the same size. I've just drawn them smaller. Okay, because I had no space. And then here is another one of these. And this will go on and on and on and on. Okay. So you have these special structures draining into this tube here. So what is this sort of a tube that's present within the submucosal layer actually called? Well, this is called a mucus tubule. Okay, so the tubes that are draining into the collecting duct here, these are mucus tubules. These tubules that are draining into the mucus tubule, these are known as serous tubules. Okay, so I think I'll go back to colour coding all of this. So I'll get my green pen for the mucus tubule here. So in green here, this portion here, this is the mucus tubule. Okay, then in blue, this portion here, this is known as the serous tubule. So what are those final structures up here called? Okay, well these structures are known as the serous acini. Okay, or for the singular is serous acinus. Okay, so this is a serous acinus. Okay, right. So the plural for acinus is acini. Okay, so you've got multiple acini here. Here's one acinus, here's another acinus. The plural is then acini. Okay, so this then is the structure of these submucosal glands. You've got these serous acini draining into serous tubules, and then these serous tubules drain into mucus tubules, which then drain into collecting ducts, which then eventually become ciliated ducts, which open up onto the epithelial surface. Now, what are these submucosal glands doing then? They are secreting the mucus. Okay? Right, so the cells of the serous acinus and the serous tubules, these are secreting the salty water that's going to make the mucus. They are secreting the primary secretion. So just like in the sweat glands where we had those coil cells which were secreting salty water, these are effectively doing exactly the same thing. There will be one important difference, however, okay, which is that the chloride channel that was being used by the coil cells, if you remember, if I get the picture back again, not that, oh, it's on here probably. Ah, that's it. The chloride channel that was being used by the coil cells was not CFTR. It is going to be CFTR in these serous acinus cells. Okay, that's the important difference, but we'll come to that in a moment. Okay, but these serous acinus cells and these serous tubule cells, they are going to be epithelial cells which are going to be secreting salty water. Water with salt in. Okay, this primary secretion, and again it's called that, a primary secretion, is then going to be draining into these mucus tubules. Now what then are the epithelial cells of the mucus tubule going to be doing? Well, they do pretty much the same as goblet cells do. Okay, these are going to be secreting mucin. Okay, and that's a horrible green smudge. Okay, but these are the mucus tubule cells, and they're going to be secreting mucin. So they're going to be putting mucin into the primary secretion to turn it into mucus. And this mucus is then going to move up the mucus tubule into the collecting duct, up the ciliated duct, and then onto the surface. And then finally, the goblet cells will continue to add mucin into that mucus. And then you've got the mucus escalator, and I don't know if I've ever written down mucus escalator, so I suppose I should put that in here. Okay, so what will happen is dirt particles and bacteria will be breathed in, they'll get trapped in the mucus, this mucus will be gradually moved up by the cilia, okay, and eventually it will get up and be swallowed, and then you keep the lungs clean this way. You're constantly making more mucus, okay, to replace the mucus that is being wafted away. And this helps to keep your airways clean and free of infection, and it is incredibly effective. Okay, right. So, what we now need to do then is we need to look at these serous acini cells, okay, in more detail. We need to look at how they are secreting this solution of salt and water, of course, okay, because it's those serous acini cells that are going to have the problem in cystic fibrosis. These are not going to be secreting as much fluid in people with cystic fibrosis, and that's what's going to drive the formation of very thick, sticky mucus, which is going to lead to the problems. Okay, right. So let's now discuss these serous acini cells. Now I'm only going to draw a little picture for this because basically we already know everything we need to know about this from discussing the sweat glands. Okay, right. So here then, let's say that this is our epithelial cell of this serous acinus here. Okay, let's say this is the apical side of this epithelial cell, and this is the basolateral side, which I'll just abbreviate as BL. Okay, so this is the side that faces into the lumen of the acinus here, and this is the side that faces out. Okay, right. So, I will remind you then of how this works. So, on the basolateral membrane, shown here as a green blob, we have the sodium-potassium ATPase, which will be establishing the sodium and potassium gradients across the cell membrane. Okay, so it will be pumping 3 sodiums out and 2 potassiums in. Okay, right. Then, also on the basolateral membrane, we're going to have the NKCC, the sodium-potassium chloride cotransporter, okay, which is going to be bringing in a sodium ion, a potassium ion, and I'm just going to make sure this is at the right height. No, it's not. Okay, and 2 chloride anions. Okay, right, and that's going to be taking advantage of the sodium gradient across the cell membrane. Okay, because when sodium moves into the cell, it's moving down its electrochemical gradient, which, remember, involves both its concentration gradient and also the electrical gradient across the cell. That will release energy, and it's that energy that we are using to move the potassium and those 2 chloride anions into the cell. Okay, right. Now, of course, we'll have a potassium channel on the basolateral membrane, which will allow that potassium to move out, so the potassium can just cycle round and round, basically. Okay, so this is a potassium channel, which I'll just colour in turquoise here. Okay, the sodium will get chucked out by the sodium-potassium ATPase. Okay, so it will cycle round and round again. The chloride then is going to get pumped out, well, moved out, not pumped out, of the apical membrane by a chloride channel. And in this case, unlike those coil cells in the sweat gland, this is CFTR, this apical chloride channel. Okay, so here is CFTR, this incredibly important anion channel. Okay, so the chloride is then going to move onto the apical face of the cell into the apical extracellular fluid, okay, or rather into the primary secretion. Okay, then it's just exactly the same thing. We have now moved chloride across the epithelial cell, okay, from the basolateral extracellular fluid to the apical extracellular fluid. That's not sustainable. We're creating negative charge over here. Okay, we've moved the negative charge from here, so we are disturbing electrical potentials. What will now happen is positively charged ions will follow the sodium, specifically sodium ions will follow from the extracellular fluid on the basolateral side to the extracellular fluid on the apical side, and then once you've moved sodium chloride, you've then got the osmotic gradient to deal with, so water again will move by the paracellular route. So sodium and water will follow the chloride by the paracellular route, and hence you have secreted an isotonic sodium chloride containing watery solution into the lumen of the acinus. Okay, right. So that now creates us a serous primary secretion. So when people talk about serous secretions, that's just water and salt basically. Mucus involves mucin in the water. Okay, a serous secretion is just salty water basically. Okay, so now the serous solution is going to come into the mucus tubules, and it's going to then have mucin added to it to create it, well, to turn it into a mucus, which will then be secreted onto the apical surface of the columnar ciliated epithelial cells of the bronchus. Okay, right, so that's the normal way that this works. So what's going to happen then in cystic fibrosis? Well, in cystic fibrosis, let's say delta F508 twice, okay, you're going to not have very much functional CFTR at the apical membrane at all, because remember, in the delta F508 mutation, the protein misfolds and gets destroyed by the protein quality control mechanisms. Okay, the tiny little bit that escapes the quality control mechanisms can end up in the apical membrane, and this actually functions okay. It's not brilliant, but it works. Okay, so you don't lose CFTR totally, but you end up with tiny CFTR function compared to what it should be. Okay, so you end up with far too little serous secretion. Okay, and then the mucus cells, they secrete the same amount of mucus. Okay, so you're putting this huge amount of mucus into a tiny little amount of fluid, and you end up with a really, really thick, sticky mucus. This is characteristic of cystic fibrosis. Thick, sticky mucus. It's just too viscous, basically. And then it doesn't really get cleared at all. So if it does manage to even get out of the submucosal gland, once it's on the apical surface of these columnar ciliated epithelial cells, it's certainly not going to get moved up in the correct way. Okay, so you break the mucus escalator, basically. In addition, most of it doesn't even get out of the submucosal glands. You end up with huge obstructions to the submucosal glands. Okay, so you end up with huge just plugs of this incredibly thick, sticky mucus in the submucosal glands because it just can't move, basically. It's just too thick and too sticky. Okay, so you lose the mucus escalator. That's the key thing. Okay, now what happens is bacteria will be breathed in, and now they don't get cleared. They stay in the lung, and this causes infection of the lung. Okay, so what I want to now talk about is what generally infects the lung first. I'll get another piece of paper for this. Okay, right. So there are two bacteria that begin the infection process generally in cystic fibrosis, and then later on it moves to a different bacteria. Okay, so when you're first born and this first starts happening, the first bacteria that generally you end up being infected with are Haemophilus influenzae, okay, which is not the cause of influenza. When it was first discovered, it was thought that Haemophilus influenzae was the cause of influenza. Later it was found that influenza was caused by influenza virus, not a bacteria at all, okay, but we didn't rename Haemophilus influenzae, but do not get confused, this does not cause influenza. Okay, influenza is caused by influenza virus. Okay, so let me give you a picture of Haemophilus influenzae just so that I have something to draw. Okay, Haemophilus influenzae is a gram-negative bacterium. It shows up red if you gram stain it. And it also has a shape that looks like this. Okay, halfway in between a round bacterium, which is called a coccus, and a rod-shaped bacterium, which is called a bacillus. So Haemophilus influenzae shape is described as a coccobacillus, okay, because it's halfway in between the two. Okay, so it's not quite a full rod, but it's not round either. Okay, so we call it a coccobacillus. So that's my picture of Haemophilus influenzae. Okay, the other bacterium that you get infected with very early on is Staphylococcus aureus, okay, which is a common bacterium that most people have on their skin. Okay, and usually it's innocuous if you breathe it in because your mucus escalator just clears it out again. Okay, but in people with cystic fibrosis it causes real problems. Now Staphylococcus aureus is a gram-positive, so it shows up blue if you gram stain it, coccus, okay, Staphylococcus. Okay, so it's a round bacterium. Okay, right. So what happens is you breathe these bacterium in, they stay in the lungs, they divide and divide and divide and divide, you get them all over the place in your lungs. What does that cause? It causes an inflammatory response. Okay, now when you get an inflammatory response in the bronchi, it's called bronchitis. Okay, so this causes bronchitis. Okay, now what does bronchitis cause? Well, firstly, you get a huge infiltration of inflammatory cells, so neutrophils, which cause the swelling of the bronchial walls, okay, and they also end up in the actual lumen of the bronchi themselves. And also, even worse, they actually stimulate the secretion of more mucus, okay, because of course the mucus escalator is the lungs' fantastic way of getting rid of infections. So the immune system has found infections in the lungs, so it thinks, right, stimulate the mucus escalator, but of course the mucus escalator is no good, it just produces more and more thick, sticky mucus, okay, and what can end up happening is you can get the actual airways themselves full of mucus, okay, so if I draw this, let me just draw a bit of the bronchial tree here, okay, smaller bronchi, and this is why it's a bit difficult because I'm drawing very large bronchi here, okay, but smaller bronchi can actually end up completely obstructed with mucus, okay, because you've produced so much of this thick, sticky mucus which you just can't clear. Okay, so I'll show it on one of these. So let's say this one's a little small one here. You can end up with mucus just filling them basically and completely obstructing the lumen. Okay, you also have to take into account that there's probably a huge amount of inflammatory exudate in here as well, so loads of immune cells that are also forming this clog, so it's just a nightmare basically. You're starting to get airways plugged up with mucus, inflammatory exudate, and also it's still going to be full with bacteria most likely as well. Right, so then what happens is it goes from bad to worse, okay? You get infected with something known as Pseudomonas aeruginosa, okay? And I don't think it's understood why it suddenly changes, okay? But initially, generally, the infections are with Haemophilus influenzae and Staphylococcus aureus. Then later on, you get infected with Pseudomonas aeruginosa. Now, Pseudomonas aeruginosa is a really horrible bacterium to become infected with. It's really, really difficult to clear. It doesn't respond very well to antibiotics at all. It's resistant to most antibiotics. There are a few that work, but it's still really, really difficult to clear. Once you are infected with Pseudomonas aeruginosa, the chances are that we are not going to be able to ever get rid of them all from your lungs ever again. Okay, so it's a horrible bacterium. Okay, so the infection moves from Haemophilus influenzae and Staphylococcus aureus to Pseudomonas aeruginosa. Now, Pseudomonas aeruginosa looks very much so like Haemophilus influenzae as far as my two descriptions are concerned, in that it is a gram-negative bacterium, so stains red if you do the gram staining process, and it is a coccobacillus. Okay, right. Now, one other thing that I want to talk about is that it causes these infections with all of these bacterium that we've just discussed also cause what's called bronchiectasis. Okay, so I want to describe what is bronchiectasis. Bronchiectasis means the permanent dilation of the bronchioles, okay, and also the bronchi. So how does dilation. Okay, so basically what happens is, if I get my picture of the bronchus back, okay, when you have this chronic inflammation of the bronchi, which is caused by these chronic infections of Staphylococcus aureus, Haemophilus influenzae, and then later Pseudomonas aeruginosa, the inflammatory cells come in, and when they fail to clear it, they get very, very aggravated, okay? You end up with a huge number of different molecules being released, okay? A huge number of pro-inflammatory molecules being released, and they all activate each other, basically. And some of these immune cells can start doing really horrible things, really dangerous things. They can start releasing very dangerous molecules that end up killing a lot of the surrounding tissue in their attempt to kill the bacterial cells. Okay, so what can end up happening is you can get the lamina propria and the smooth muscle cell layer starting to deteriorate. Okay, so you lose a lot of the smooth muscle cells, and you lose a lot of the elastic tissue that is within these connective tissue layers, such as the lamina propria. Okay, and when you lose those smooth muscle cells and those elastic tissues, of course what's going to happen is the whole thing's going to dilate. The smooth muscle cells were responsible for constricting the bronchi down. The elastic tissue was also responsible for constricting it down. When you lose those, the whole thing sort of sags apart, okay, and ends up hugely dilated. Okay, so that's what is meant by bronchiectasis, this permanent dilation of the bronchi that is because the smooth muscle cells and also the elastic tissue of the bronchi has been destroyed by the inflammatory response that is being launched because of these chronic bacterial infections, basically. Okay, right. So then the final thing that I need to tell you is, how does this eventually lead to death? Well, basically, we've discussed how bronchi can end up completely obstructed. Okay, when enough of your bronchi and enough of your bronchioles are completely obstructed, you can no longer use your lungs adequately. Okay, when you've obstructed a bronchi like this, all of the bronchioles and all of the alveoli downstream of this, so I'll draw a huge great segment of the lung here, all of this is now not going to get oxygen coming into it. So all of these alveoli are now non-functional, basically. They're not helping to bring oxygen into the blood or expel carbon dioxide. When enough alveoli are no longer useful, okay, then your lungs are not going to be able to function anymore. They're not going to be able to do their job of bringing in adequate oxygen to supply the tissues of the body and expelling the carbon dioxide which the tissues of the body are producing, okay? And that's when the body dies, okay? And when you die because of the lungs failing you, that's known as respiratory failure or respiratory insufficiency. Okay, right, and that's the most common cause of death in cystic fibrosis nowadays. Okay, right, so that's then the respiratory symptoms of cystic fibrosis. I think we'll call it there for this video now, and in the next video what we will move on to is the pancreatic symptoms of cystic fibrosis, and we'll explore why cystic fibrosis is called cystic fibrosis. We'll see the cysts and the fibrosis within the pancreas.